Welcome to the SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. In the last few lectures we have seen the microbial growth, and in that we have seen the microbial growth characteristics, the nature of microbial growth curve and different factors affecting the microbial growth, and we have seen also the bacterial measurement of bacterial growth. In this lecture we will be seeing discussing the food spoilage, and let us discuss first overview of the food spoilage, and we will discussing in this lecture the introduction, major causes of spoilage, classification of foods based on spoilage, factors affecting kinds and number of microorganisms in food, factors affecting the growth of microorganisms in food, sequence of events in food spoilage, and lastly the chemical changes caused by microorganisms. Food spoilage is defined as a microbiological, chemical or physical changes in food items which renders a product undesirable or unacceptable to the consumer for consumption. In other words, food spoilage can be defined as any visible or invisible change which can make food or product unacceptable for human consumption. Microbiological food spoilage is produced by the growth and action of microorganisms in food. It is because of the production of objectionable end products due to biochemical activity of microbial chemical processes. Chemical spoilage may be caused by the non-microbial enzymic action, oxidation, or because of some processing problem like non-enzymatic browning. Spoilage may also be observed because of different components of food react with each other or with some component added or mixed during manufacturing process which alter the food's sensory characteristics. Physical food spoilage includes water loss or dehydration, increase in moisture or absorption of excessive moisture, freezing burn, and recrystallization of frozen foods. About 25% of total food product produced is spoiled due to microbial activities. Major causes of food spoilage. The major causes as enumerated here, like the reaction with oxygen and or light, the time, the physical stress or abuse, inappropriate temperatures, infestation by insects, parasites and rodents, or food enzymes and other chemical reactions within the food itself, or microorganisms like bacteria, yeast and molds, and gain or loss of moisture. All these cumulatively deteriorate the food. Spoiled term is applied to food when there is decay or decomposition of undesirable nature found or observed in food, while food unfit for consumption due to sanitary reasons is not called spoiled. During spoilage, the original nutritional value, texture, flavor of the food are damaged and food become harmful to people and unsuitable to eat. The spoilage may be due to one or more of the following, like insects, action of enzymes of the plant or animal food, purely chemical reactions, for example browning, staling, rancidity, etc., that is non catalyzed by enzymes of the tissues or of microorganisms. Physical changes such as those caused by freezing, burning, drying, pressure, etc. or it may be due to growth of activity of microorganisms. The microbial spoilage does not appear to develop gradually day by day, a little worse, but more often as an unexpected and unpleasant revelation. This is due to exponential nature of microbial growth, and as a result metabolic activity also increases with an exponential rate. This food spoilage, that a pictorial representation of food spoilage, it is caused by mainly moisture loss, enzyme action, microbial contamination. The moisture loss is due to once harvested fruits and vegetables cannot absorb moisture from the soil, which results in shrinkage, a limp appearance. The enzyme action, that is because of enzymes are naturally present in food. They are involved in food spoilage as they cause ripening of the food. Certain enzymes present in fruits and vegetables cause ripening. As enzymes ripen, changes occur: taste, color and texture. Enzymatic browning: some fruits and vegetables, for example apple, releasing the enzyme oxidase, which when cut, oxidase reacts to oxygen in the air, turning the food brown. Or enzymatic deterioration: the enzymes naturally present in fish cause deterioration even at low temperatures. Therefore, fish must be consumed or processed quickly. The last one is the microbial contamination. The microorganisms are the principal cause of food spoilage. Food can provide the ideal conditions for growth. The main microorganisms responsible for food spoilage are yeasts, molds and bacteria. When yeasts and molds they grow, they cause the visible food spoilage. Generally, they do not cause food poisoning, but some molds can produce mycotoxins which can be a health risk, whereas the bacteria, they cause invisible food spoilage on the exterior and interior of the food, commonly cause of the food poisoning. Now, the classification of foods based on spoilage. The food may become unacceptable to the following factors: growth and additives. Growth and activities of microorganisms, principally bacteria, yeast and molds. This is by far the most important and common cause of food spoilage. Activities of food enzymes, that is enzymatic browning, is a common example. Infestation by insects, parasites and rodents. Chemical changes in food, for example, the chemical changes in food that is not catalyzed by enzymes of the tissues or of microorganisms. For example, the chemical oxidation of fats producing rancidity, as well as non-enzymatic browning reactions in foods like Maillard browning. Physical changes or damages such as those caused by freezing, that is the freezer burn, by drying, that is caking, etc., or presence of foreign bodies. Physical abuse, that is contamination with chemical agents. Thus, all types of spoilage changes can be broadly categorized into physical, chemical and microbiological types. On the other hand, the food can be categorized into following three groups: stable or non-perishable foods, foods which do not spoil unless handled carelessly, for example sugar, flour and dry beans, etc. Semi-perishable foods, foods if handled and stored carefully remain unspoiled for fairly long periods, for example potato, apples, nut meats, etc. Perishable foods. Daily foods that spoil readily unless special preservative methods are used. For example, meat, fish, poultry, most fruits and vegetables, egg and milk, etc. This is the pictorial representation of different types of food. Like non-perishable foods, they stay good up to an year. For example, sugar, legumes, oils, pickles, etc. when stored in a cool and dry place. The another type is semi-perishable foods which stays good for a week or few months and includes semolina, gram flour, onions, potatoes, apples, frozen foods, etc. if they are stored in ventilated cool room or fridge. The last one is the perishable foods. They spoil within a couple of days and include milk, eggs, meat, fish, poultry and most fruits and vegetables, especially green leafy vegetables. Now, the factors affecting kinds and number of microorganisms in food. Environment in which food is kept or come from, that is soil, air, etc. The post contamination, growth characteristics of microorganisms, pre-treatment of food before storage. This picture reflects the factors affecting the growth of microorganisms in food. That food systems, they inherently contain carbohydrates, protein, fatty acids and ions and minerals, which form the suitable platform for bacterial fungal infestation and toxin secretions. The bacterial toxins are generally produced by Clostridium, Enterobacter, Salmonella, Bacillus and Vibrio species. They produce exotoxins, they produce enterotoxins and heat labile toxins, whereas the fungal toxins, they are produced by Aspergillus flavus, Aspergillus parasiticus, Aspergillus candidus, Clostridium, Cladostorium, Cladosporids, Alternaria alternata, Fusarium oxysporum and Fusarium verticuloides, Fusarium verticilloides, and they produce alpha toxins, fumonisins, zearalenone, deoxynivalenol, ochratoxin, botulin and trichothiazine. And the factors affecting the bacterial toxin productions are environmental factors which include the temperature, rainfall, relative humidity and maturation periods, whereas for fungal toxin production the abiotic factors include pH, moisture content and water activity, whereas the genetic factors include transcriptome, metagenomics, sequencing, genetic resistance and hybrid vigor. The associative growth. Competition between different kinds of bacteria, yeast and molds. If conditions are favorable for all, then bacteria will grow faster than yeast. Yeast will grow faster than molds. That is, bacteria is faster than the yeast and yeast growth is faster than molds. Among the bacteria or yeast or molds also there is a competition. Microorganisms are not always antagonistic or antibiotic to each other. Many times they may have symbiotic, that is mutually helpful or live and let live basis. They may grow simultaneously without seeming to aid or hinder each other. Two kinds of microorganisms may be synergistic, that is when growing together they may be able to bring about changes, for example, fermentation that neither could produce alone. For example, in milk, if the Pseudomonas cincinnati alone will produce only a light brownish tinge. Streptococcus lactis, when alone, will produce no color, and when grow together they produce bright blue color. The metabolic products are metabiosis. When one organism makes conditions favorable for growth of the second, the two organisms may be growing together, but one succeeds other and makes the conditions favorable for the second. For example, most natural fermentation are the decomposition of raw milk, which is represented in this figure. The raw milk, if it is kept at room temperature, then the Streptococcus lactis and coliform, they will start to grow, and as a result of their growth, the acid produced, and after sufficient acid is produced by these two organisms, their growth will be inhibited, and then the lactobacilli will start to grow and produce more acids and makes the pH low, and again they get inhibited. After that, the yeast and mold will grow and reduce the acidity to neutral level, and then proteolytic bacteria will grow, which will ultimately result in liquefaction of the proteins. Environmental conditions. This includes physical and chemical properties of foods that favors or retards growth of certain organisms. Acidity, sugar, water activity, etc., presence of antibiotic substances or inhibitors, or availability of oxygen, temperature. This is not contained, this is certain. Sequence of events in food spoilage. Microorganisms have to get into food from a source or more. Food environment should favor the growth of microbes. Food need to be stored under the growth condition for a sufficient length of time. Now, the chemical changes caused by microorganisms. Changes in carbohydrates. The carbohydrates are used to obtain energy, while monosaccharides are preferred over complex carbohydrates. Microorganisms have ability to convert polysaccharides to simpler forms before obtaining energy. The utilization of simple sugar such as glucose vary under aerobic and anaerobic conditions. In aerobic conditions, it is converted into carbon dioxide and water through glycolysis and other related pathways. In absence of oxygen, the process yields a number of compounds in different organisms. This process is known as fermentation. These compounds include alcoholic fermentation, for example, by yeast with production of ethanol and carbon dioxide. Simple lactic fermentation, for example, by homofermentative lactic acid bacteria with lactic acid as main product. Mixed lactic fermentation, for example, by heterofermentative lactic acid bacteria with production of lactic acid, acetic acid, ethanol, glycerol, carbon dioxide as main products. Coliform type of fermentation, for example, by coliform bacteria with production of lactic acid, acetic acid, formic acid, ethanol, carbon dioxide, hydrogen, and also acetoin and butanediol like products. Another is propionic fermentation, for example, by propionic acid bacteria with production of propionic acid, acetic acid, succinic acid and carbon dioxide. The another one is butyl and isopropyl fermentation by anaerobic bacteria yielding butyric acid, acetic acid, carbon dioxide and hydrogen. This figure shows the type of carbohydrates, especially selects microbial community structures and fermentation patterns. Like the mixed fermentation, if it is there in the medium, then the C6 carbon polysaccharides is preferred by bacilli, followed by the Clostridia, and this is lactate and the hydrogen will be produced. Whereas the C5 carbon atom polysaccharides are the sugars that will be preferred more by the Clostridia than by bacilli, and butyrate and hydrogen will be produced. Whereas if the polysaccharides are present, then that will be preferred by Ruminococcaceae family, and hydrolytic activity they will perform to produce the simpler sugars and hydrogen. Changes in nitrogenous compounds. The proteins are the major source of nitrogenous compounds in foods, and its degradation through hydrolysis or enzymatic reactions by enzymes either produced by microbes or enzymes naturally present. Proteins are converted into polypeptides, peptides, amino acids, amides, amines, etc. The enzymes proteinase involved in conversion of proteins into polypeptide, while peptidases catalyze conversion of polypeptides to amino acids. This decomposition process are either aerobic or anaerobic. Anaerobic decomposition of proteins produce obnoxious odors known as putrefaction. Nitrogenous compounds with some other compounds are responsible for such smells, include sulfur compounds. Amino acids cause either deamination, that is removal of amine group, or decarboxylation, that is removal of carboxyl group, by the action of microorganisms. The other microorganism involved in conversion of nitrogenous compounds include Pseudomonas, E. coli, Clostridium, D-sulfotomaculum, etc. This is the pictorial representation how protein gets disintegrated. The protein, it is acted upon by proteinases to peptides. These peptides are acted by the peptidases to produce simple peptides and again ultimately to amino acids. These peptides and simple peptides, they give bitter taste to the foods, whereas the amino acids, they give flavor to foods whether it is desirable or undesirable. For example, amino acids contribute flavor to the ripened cheese. Anaerobic decomposition of proteins, peptides or amino acids result in production of obnoxious odor and is called putrefaction due to production of sulfur containing products, for example H2S, methyl and ethyl sulfides, mercaptans, which is another sulfur containing compounds, ammonia, amines, for example histamine, tyramine, piperidine, putrescine and cadaverine, indole, skatole and some fatty acids. When microorganisms act upon amino acids, they may deaminate, decarboxylate or both. Depending upon the E. coli and the substrate, the different products are formed. For example, when E. coli acts on the glycine amino acid, then glyoxylic acid, acetic acid and ammonia is formed. When Pseudomonas acts on the glycine, then methylamine and carbon dioxide is formed. When Clostridia is act on the glycine, then acetic acid, ammonia and methane is formed. and E. coli, when the amino acid is changed, that is the alanine, then alpha keto acid, ammonia and carbon dioxide is formed. Whereas the Pseudomonas, when acts on the alanine, the acetic acid, ammonia and carbon dioxide is formed. Whereas the Clostridia, when act on the alanine, propionic acid, acetic acid, ammonia and carbon dioxide is formed. Changes in organic acids. The organic acid is decomposed aerobically by film yeast to carbon dioxide and water. Acids may be oxidized to either simpler acids or products similar to those for sugars. Organic acids, which are generally present in form of salts, are oxidized to carbonates, which makes the medium more alkaline. Saturated fatty acids or lower ketone derivatives are degraded to acetic acid, that is 2 carbons at a time, with the help of coenzymes. Unsaturated or hydroxy fatty acids degraded in the similar manner, but must be converted to a saturated fatty acid or ketonic derivatives for complete oxidation. Changes in lipids. A number of microorganisms produce lipase enzymes causes hydrolysis of lipids, and the major end products are glycerol and fatty acids, which will be further utilized by those microorganisms for their metabolism. The oxidation of fats is done by inherent enzymes of food, and mostly high fat containing foods have this type of defect. Now, changes in pectic substances. Protopectin, which is water insoluble and parent pectic substance of plants, is converted to pectin, which is a water soluble polymer of galacturonic acid, which contains methyl esters, ester linkages and varying degrees of neutralization by various cations. It forms gel with sugar and acid. Pectin esterase causes hydrolysis of the methyl ester linkage of pectin to yield pectic acid and methanol. Polygalacturonogens destroy the linkage between galacturonic acid units of pectin or pectic acid to yield smaller chains and ultimately D-galacturonic acid, which may be degraded as simpler sugars.